In this section, we will talk about triplet code. There are 4 nucleotides or bases in deoxyribonucleic acid, which are adenine, cytosine, guanine and thymine. But genetic code is a combination of 3 nucleotides, which specify a particular amino acid. There are 3 nucleotides in a codon, for example, adenine, cytosine and guanine, and we have discussed that. The genetic code is a triplet code, and the reading occurs continuously without punctuation between the 3 nucleotide units. Moving forward, 1 nucleotide codon would not yield enough to code for the 20 different amino acids that commonly occur in proteins. Let's understand that. Why codon is combination of 3 nucleotides? Because 4 into 1 equals to 4. And a codon with 2 nucleotide would not yield enough combinations to code for the 20 different amino acids that commonly occur in proteins. Because 4 into 4 equals to 16. But a codon with 3 nucleotide would yield enough combinations to code for the 20 different amino acids that commonly occur in proteins. Because 4 into 4 equals to 64. That's why a codon with 3 nucleotide would yield enough combinations to code for the 20 different amino acids that commonly occur in proteins. Moving forward, after Crick's initial experiments, Marshall Marenberg, Philip Leader, and Har Gobind Khurana tested all the 64 codons by making artificial messenger ribonucleic acids and triplet codons and using them to synthesize a protein. But, as we have discussed that, out of 64 codons, 3 codons, uracil, adenine and adenine, and uracil, adenine and guanine, and uracil, guanine and adenine, do not code for any amino acid, and hence are known as nonsense codons. So, every gene starts with initiation codon, adenine, uracil and guanine, which encodes the amino acid methionine, and nonsense codons are usually present at the end of the gene, and hence are also called stop codons. In some cases, the genetic code is universal. It is the same in almost all the organisms. For example, adenine, guanine and adenine specifies arginine. In bacteria, in humans, and all other organisms, whose genetic code has been studied. Because of the universality of codon, the genes can be transferred from one organism to another, and be successfully transcribed and translated in their new host. But, as we have discussed that, some cases are exceptional. The study of genetic code of mitochondrial deoxyribonucleic acid, however, showed that genetic code is not that universal. For example, uracil, guanine and adenine codon is normally a stop codon, but in mitochondria it reads as tryptophan. Likewise, adenine, uracil and adenine was read as methionine instead of isoleucine, and adenine, guanine, adenine, and adenine, guanine, guanine, for termination of protein synthesis, is instead of arginine. Thus it appears that genetic code is not quite universal. So, this was all. In our next section, we will talk about the frame shift mutation.
Have you ever heard of video books? sqadia.com is delighted to present video books, medical books that have been tailored into a series of videos. sqadia.com videos are presented in the same order as the book's chapters. Explore now and make your learning easy.